So I have a brief discussion on heterocycles. Many biological molecules, many pharmaceuticals have a nitrogen-containing aromatic heterocycle. Here's a coenzyme, right, common in metabolism, I believe, something like that. Here's the structure of Viagra. Has 1, 2, 3 nitrogen heterocycles. 2 of them are aromatic, right? This one is not, just a 6-membered ring. But look at heme right here, the compound that allows us to breathe oxygen, right? Kind of important. It has this nitrogen heterocycle structure that captures an iron 2 ion, complexes it in the middle of it, and that's what allows oxygen to be carried by your red blood cells. So the 2 aromatic heterocycles we talk about in here are pyrrole, right? That's the aromatic 5-membered ring with an NH in it. So it's got 2 double bonds, CC double bonds, and then an NH as the 5th unit. The nitrogen in pyrrole is not basic. Okay? It has a lone pair, but that lone pair is part of the aromatic system. Pyrrole only has 2 CC double bonds in it, so that's 1, 2, 3, 4 electrons. Right? Imagine those. To get up to the magic 6 number, we need to include nitrogen's 2 electrons within that pi system. So nitrogen's lone pair is in a unhybridized p orbital that's lined up with all the other pi bonds. pKa of 0.4 means the conjugate acid of pyrrole is essentially a strong acid. And that's such a strong acid because protonating pyrrole would mean that we would have to break aromaticity. Right, this compound NH2 plus is not aromatic anymore. Very strong acid because removing that H gives us an aromatic, very stable compound. Pyridine, however, has a basic nitrogen. Pyridine is just benzene with an N in it, and it's a 6-membered ring, has 3 double bonds in it, so it's got all 6 of its electrons that it needs to be aromatic. The nitrogen lone pair is sticking out. It's not aligned with all the other p orbitals. It's in a hybrid orbital that's sticking straight outwards. So that's available to go off and get protonated, right? The pyridinium ion is still aromatic. So we don't lose any stabilization or anything, really. The pyridinium ion has pKa of 5.25, right, which is a weaker acid, which means pyridine is actually basic. In fact, just as a reminder, that's why we use pyridine. It has about the right pKa to react with excess HCl that's formed in those acid halide reactions. Right? If I take an acid halide and want to make a ester out of it, we have pyridine there to neutralize the HCl that's produced. So I've got a couple other ones for you to look at. Let's look at imidazole. It's a 5-membered ring. It has 2 nitrogens in it. One nitrogen is double bonded in the ring. Right? We call that a pyridine-like. Right? Because it's an N that's double bonded within the ring. The other is like pyrrole. It's an NH. So there's a lone pair right there. Only one of those nitrogens is basic, however. If I draw this from the side, hopefully we'll be able to see a little bit how this is going to work. Let's draw p orbitals on all these atoms because imidazole is aromatic. So it has to have p orbitals on each atom. The double bonded nitrogen is right there. It has a lone pair that is going outside of the ring, right? It's not participating. Whereas the NH, its H is pointed outside of the ring, and its lone pair has to participate in the aromatic system. So the pyridine-like nitrogen that has its lone pair pointed outwards is the basic one. Take a look at just a couple of these polycyclic heterocycles. Polycyclic just means we have multiple rings that are fused together. So quinoline is 2 aromatic rings, one of them has a nitrogen in it. Isoquinoline just has a nitrogen in a different place. Indole has a 6 and a 5 membered ring with the nitrogen in the 5. Purine has 4 nitrogens in it, 2 in each ring. So those basic building blocks, I suppose, are common in many, many biological molecules. So look at quinine, the compound that revolutionized the treatment of malaria, for instance, in Africa. Quinine has a quinoline core to it. Tryptophan is an amino acid that has an indole ring in it. Look at adenine. Okay? Those of you who've been through biology, you know that adenine is a very important constituent of DNA. It has purine at its very core. It's purine plus an additional NH2 bonded to the ring. Real popular molecule, I'm sure, with a lot of you is caffeine. Let me draw caffeine for you. Caffeine has a core very similar to adenine. The double bond here to this N is in slightly different places, but it has 2 carbonyls in the rings, and each single bonded nitrogen has a methyl group. Double bond right there. That's caffeine. Very similar to adenine in its structure. You're probably saying, well, that's not so much the same. Those nitrogens are single bonded in the ring, not double bonded. But if I have C double bond O with an N next to it, I could imagine this becoming an OH, putting a double bond in the ring. That's sort of like if this is keto, enol would be like this with the double bonds within the ring that way. So sort of like the keto tautomer of an adenine substituent.